In this experiment, we are going to determine initial rate of the cellulose enzyme that degrades cellulobiose. For it, we would need cuvettes, stop solution, DI water, substrate at 1.5 millimolar concentration, resuspension buffer, and enzyme. For this activity, we will also need the spectrophotometer, pipettes, and pipette tips. Take the spectrophotometry cuvettes and fill one with 500 microliters of stop solution. Stop solution is a strong base. Add 400 microliters of 1.5 millimolar substrate and add 100 microliters of resuspension buffer. Gently mix and set aside, labeling it with the C letter. It is going to be our control cuvette for calibrating the spectrophotometer. Now add 500 microliters of stop solution into each of the remaining cuvettes. Now we are going to set up reaction, and for that we will add substrate and enzyme to 15 ml conical tube. So we first add 2 ml of 1.5 millimolar substrate and then add 1 ml of enzyme. Following enzyme addition, we have to count time, because the enzymatic reaction is time sensitive. So for this activity we will need a timer. As far as we are going to measure amount of p-nitrophenol formed at 1, 2, 4, 6 and 8 minutes, we are setting the timer for 8 minutes. Now we are adding 2 ml of 1.5 millimolar substrate into 15 ml Falcon tube. Followed by addition of 1 ml of enzyme. We set up the timer. When 1 minute passed, transfer 500 microliters from reaction mixture into the cuvette number 1. When 2 minutes passed, take 500 microliters from the reaction mixture and transfer it to the cuvette labeled 2. After 4 minutes, take 500 microliters and transfer it to the cuvette labeled 4. At 6 minutes, take 500 microliters and transfer it to the cuvette labeled 6. When the timer is over, transfer 500 microliters into cuvette labeled 8. Now all cuvettes are ready and we start the absorbance measurement. First we are going to blank our instrument with a control sample labeled C. So by placing the cuvette and pressing calibration button, we are going to zero the measurement. Now take the absorbance measurement from the cuvette labeled 1. Repeat the reading for cuvettes labeled 2, 4, 6, 'and the last cuvette labeled 8. In order for your data to be reliable, you need to repeat the experiment one more time. Of course, the control cuvette will be different from the previous set of the measurements. Increasing the number of replicates of the experiment will give us the possibility to average the measurements and obtain most reliable results. Here you could see the values of the measurements of the absorbance for the run 1 and run 2. Now you need to take the average and obtain the reliable results. Using standard curve you built earlier, determine the concentration of p-nitrophenol at each time point, and then determine the initial reaction rate for this set of the experiment.